Hello, audible? In the chilled wintry morning, all of us are cuddled in under our blankets and wondering whether should we go out for bird watching or not. Isn't it an always been a very extremely difficult question for all of us who are bird watchers here? And I ask myself, because it's cold outside, why would there be a birds hanging around? Very often when we go out of our houses, we look at forest. It's dead silent in the morning. But often we see that there is some buzz in the corner, and we wait a little, and before we realize anything, there is a whole lot of activity, and we are surrounded by a lot of birds, making lot of noise, right? And they're just like a wave. They come from some one direction, they'll pass through in front of you, and they'll again disappear into the forest. And this phenomenon is extremely common throughout the world in all terrestrial habitats, and they're popularly known as the hunting parties or the mixed species bird flocks. And birds in these flocks have different foraging styles, and since they move throughout the day, they flush lots of insects from all sides, all corners of the forest, and make them available to all fellow participants and help them survive the harsh winters. And this situation is even more far contrast up there in the mountains because of crazily cold climate. So this is what I really got onto and ended up working as a study for my master's dissertation on species interactions with species bird flocks along an elevation gradient in the Western Himalaya. Western Himalaya in particular lies latitudinally much higher than other mountain ranges in India, and it provides a perfect test to check the responses of flocks to harsh climatic conditions in the higher elevations to the lower upper Shivalik habitat, which is relatively much more drier and warmer. And before I even went on to do this study, I had a few objectives and associated hypotheses. So the one major objective was to understand the flocking propensities and interspecific interactions between this species along an elevation gradient. And since along this elevation gradient, the resource availability is expected to decline because of harsh temperatures, I expected that the association strength between these species, their interspecific associations, as well as their flocking propensities, which is the tendency of species to take part in the species flocks, will increase with increasing elevation, so that they can avail the foraging benefits of participating in the mixed species bird flocks. And I expected the same thing to happen in summer, because in summer still the high elevations are relatively colder than the low elevations. So I worked right here in our backyard, starting from Rajati Tiger Reserve in the Dehradun district as a low elevation site, to the high elevation site in Mussoorie, on the Mussoorie Ridge. So I divided this entire gradient into 3 elevation bands, the low elevation band being in the foothills of Shivalik. The mid elevation band was close to 1200 meters near the northern boundary of Dehradun Valley at the base of Lesser Himalaya, and the high elevation band was on the ridge of Mussoorie. And these circles represent my trails. So each field site had two trails, and these trails were around 3 kilometers in length. So I sampled mixed species bird flocks as well as relative abundances of birds along these 3 existing trails, always in the morning, within 10-meter strip width on both sides. So effectively speaking, I walked the trails and recorded everything within as well as outside flocks in 20-meter strip width. I also looked at arthropod availability by using bagging method, where I cut the branches, put them in the polythene bag, and then assess the arthropod availability. I saw a lot of variation in the body sizes of birds. So in order to see whether large-bodied and small-bodied birds flock separately, I did a cluster analysis on the species composition of flocks. I calculated flocking propensities by dividing species incidence within flocks by total incidence inside as well as outside flocks. And I used a paired t-test to compare the flocking propensities between 2 adjacent elevations. Since my studies spanned across 2 seasons, winter and some part of summer, I delineated flocks using average flocking propensities of the 4 species. To study the interactions within these flocking birds, I constructed networks. For that, the association strength, which is also known as a weighted degree, was calculated based on the co-occurrences of two species in a flock. So if two species co-occurred less, then they have very low association strength. If their co-occurrence is more in the flock, then the degree or the association strength is very high. I also calculated something known as network density that tells us the actual connection, actual associations in a large network. So suppose we have four species here, all possible interactions within these four species will be 6. But actually if you look at here, there are just 4 major interactions happening, and these two, the blue dotted lines, are absent. So the network density becomes 4 by 6, which is 0.66. But there is a major problem here. Species will interact more or less with other species based on their relative abundances in an area. If species is very common in the area, it will be associating with many species. It will be present everywhere. It will be there in all flocks. So to control for the availability of species, I simulated null model and I randomized that process. So the simulated flocks were made in proportion to the relative abundances of birds. So I sequentially filled species in the flock in proportion to their relative abundances, where the flock size, the entire species pool, and the number of flocks for each iteration was kept constant as the observed flocks. And after iterating it 10,000 times, I have a distribution of 10,000 null values that are represented by this box plot. And the colored portion of box plot represents the 95% confidence interval. Then I compared the observed value, which is coming from my field data, to the null distribution. And I compared whether the observed value falls within 95% confidence intervals, below or above. If it is below that, then species are associating less than expected. If it is above that, then the species are expecting more than expected. And if it's within that, then species are associating as expected. I also calculated z-scores by subtracting the mean of null from the observed value and divided it by standard deviation. The association strengths were compared across two elevations because two elevations will have more common species, and they were compared in the pairwise fashion. The realized interspecific interaction is a network density. It's a network level property and was compared with the help of a single z-score. So I had 3 z-scores for 3 elevations. So it was direct comparison. And to analyze arthropod diversity, I plotted number of arthropods per batch against elevation, as well as number of insects per batch against elevation. And to check whether the diversity of arthropod changes with elevation or not, I quantified the Salmon diversity index. It was actually fun working in the mountains because of the snow, and I had to actually wade through snow many times. I had to actually drive by on the snow, and it was fun walking the trails just after the snowfall, full of bird activity. And I walked 72 trails, which equal to almost 210 kilometers. And I observed 18 species participating in 412 flocks, of which 72 flocks were in the low elevation, 156 were at the mid elevation, and high elevation I recorded 179 flocks. So the differences in these sample sizes were mainly because of the flocking period itself. Flocking period was very short in the low elevations, and it progressively increased with increasing elevation. So low elevation had just 2 months of flocking or 2.5 months of flocking, whereas high elevation continued to flock more than 4 months. To delineate flock data across 2 seasons, I used flocking propensities of 4 species. And I found that the average flocking propensity is somewhat decreasing in the mid-March. So I used mid-March, 15th of March as a boundary to separate between 2 seasons. And I treated all flocks before 15th of March as winter flocks and after 15th of March as summer flocks. In the high elevation, I found 2 flock types, mainly categorized by their species composition being very different because of the body sizes of birds. So here, each branch of this dendrogram represents a flock, and these flocks are clustered based on their species composition. So the flocks highlighted in red were significantly of large body size birds, more than 40 gram body weight, and a large chunk of flocks below are of small bodied birds. So I did not analyze the large bodied bird flocks because of the low sample size, almost 15 flocks, but I analyzed all other small bodied bird flocks. And for the low and mid elevation, there were no different flock types, just only small bodied bird flocks. Then I looked at flocking propensities of species, which is actually the tendency of species to flock. It's an actually innate property of species whether to flock or not, and it changes with elevation because of differences in the resource availability. And I found that there is no really significant change between low and mid elevation in the flocking propensities. The pairwise comparison by using Petri test did not show any significant results. But on the other hand, if you look at for the mid and high elevation, there is significant increase in the high elevation flocking propensities. They are always close to 1. So which says that species are always in the flocks. They don't want to move on their own. They always participate in the flocks. But whereas mid elevation species can forage on their own without even participating in the flocks. In summer as well, I found high elevation to have continued high flocking propensity than mid elevation. The low elevation data was not analyzed because flocks disintegrated even before I started sampling for summer. Propensities across seasons also changed. For the mid elevation and low elevation, both had low propensities in summer than in winter. Apart from the flocking propensity, I also looked at interspecific associations of these birds. And I found that they also respond to elevation. So here, basically, this box plot is representing a null distribution of a network value called network density. And the black dot here, box plot is null distribution, and the black dot is an observed value. So here we have to basically compare the black dot observed value with null expectation. So this box plot are basically compared vertically but not horizontally. So if you look at the distance between black dot and the mean of this box plot, it's pretty high if you compare to mid and low elevation, mid and high elevation. So as you go along the increasing elevation, the observed value is getting closer and closer to null expectation, suggesting species are participating, species are interacting in high elevation as expected from the null expectation. But whereas at the low elevation, species are not expecting as their null expectation because maybe there is enough food available or they don't want to simply participate in the flocks because they can very well forage on their own. So this graph shows the construction of network across 3 elevations in both seasons. Here the circles representing species, and the size of circle tells you how strongly the species is associated with other species. And the width of this line tells us the association strength between those species. So if you look at from the bottom, this is for the low elevation, the Z scores are actually increasing with increasing elevation, suggesting the same thing. The observed value is getting closer and closer to the null value. That's why the Z scores are increasing. And across seasons, Z scores are decreasing at any given elevation. And if you look at for the low elevation in summer, the flocks are completely disintegrated, largely because of the upward movement of birds and maybe because of the more food availability. I also looked at something known as weighted degree that tells us the association strength between participant species. So when compared in winter, I found associations tend to increase with increasing elevation irrespective of any elevation band. In summer as well, the winter had the highest association strength than mid elevation. And across seasons, the association strength only decreased for the mid elevation but not for the high elevation, probably suggesting that there is still not winter, still not summer arrives in the high elevations, but mid elevation flocks started to disintegrate and they have low association strength. So interestingly, when I looked at arthropod availability across this elevation gradient, I sampled 150 bags, in which 50 bags were sampled at each elevation. And I classified these arthropods into 13 insectoids and spiders. And interestingly, I did not find any difference in the arthropod availability. Arthropod availability was more or less similar. All box plots and error bars completely overlap. But this is largely because there is high relative abundance of spiders in the higher elevations. Probably something to do with moisture, because in the low elevation I did not find much spiders. Maybe because of its shivalic habitat, it's much drier. Spiders are higher there in the high elevations. But when I separated spiders from the overall arthropods and just plotted only insects per bag, excluding spiders, I found a trend which shows that the insect availability is actually decreasing with increasing elevation. And this also supports our theory that flocks have loose connections or loose associations in the low elevation because of more availability of food, and in the high elevation because of less availability they have very strong associations. I also calculated salmon diversity index, and it was almost similar for the low and mid elevation. This was calculated on the arthropods, but significantly low for the high elevation. And arthropod availability was not assessed for the summer season, it was only done during the winter season. To conclude, even though my studies spanned across just 4 months, it did give some insight on how flocks respond to elevation gradients and why do bird flocks at all at different elevations differently. So I feel that flocks do respond to these gradients largely because of differences in the resource availability or the food availability. When there is a resource crunch, species try to help each other and try to facilitate each other by participating in the flocks and exploit the flocking benefits. With increasing elevation, species don't just flock more, but they interact with many more species, and these associations, these interactions become stronger than null expectation as you go with increasing elevation. Null models are extremely crucial to understand any social network studies when there is abundance playing role. So you have to compare for the abundance of species, and then the true picture of interactions can be better understood. In future, study can focus on functions of nuclear species, which are very, very important in these flocks, which I couldn't do because it needs a lot of observational data. You have to follow flocks continuously for the day, and how their roles change with increasing elevation. This is something that can be studied in future. Responses of flocks to varying predation pressures. So apart from the availability of resources, predation pressure is major driver, and flocks are known to respond to predation pressure. So this is something can be explored across varying predation pressures, maybe across elevation or across different landscapes. And propensities can be controlled with an incidence-based null model to study the effects of propensity and interspecific interactions separately and how they respond to the flocks. And with this, I wish to thank all these people. Without their help, this study wouldn't have been possible. And thank you.
Can we have a question?
Yeah.
Pranav, very interesting study.
Thank you, sir.
Just one minor query. So as the elevation goes up, there is less resource availability, and then that is why there is high flocking, mixed species flocking.
So actually we don't know what species feed on what resource.
Yes.
So when I looked at all arthropods together,
Yes.
it seems like there is no difference at all across elevations.
That is why my question is. So when you look at all the arthropods, there is no difference.
There is no difference.
But when you remove spiders, and then there is a trend that you have seen. So, but my question is, do these birds feed on spiders?
Birds are known to feed on spiders, but primarily all of these birds, these flocks are insectivorous. But they do feed on spiders, they're known to feed on spiders. But we don't know at what proportion they feed. Do they feed only on spiders or I mean relatively more on spiders or relatively more on insects? That we don't know.
So what do other studies say, like for example in other landscapes?
So far nobody has really looked at how flock respond to resource availability. They actually quantifying resource availability. So there is no direct comparison anywhere. So people have just predicted that with increasing elevation, generally we know that the resources will be less up there, and that's why you have flocking more strong. But if you quantify them, then the picture will be more clear.
Maybe in future, maybe.
Yeah.
Very interesting study.
Thank you, sir.
Pranav, it was really delight to listen to your talk. Very
Thank you, sir.
interesting
Thank you,
study.
sir.
So my first question, you have already put in your future. So I was interested in looking that are there some species which act as a nodal species,
Yeah.
and do these nodal species change with elevation?
So So,
Right.
so I do not have data to say that, but from the natural history observations, what I feel that the ticks are very important in this class. The scenario tick that we commonly see in the campus. If you go high, there is greenback tit, black-throated tit, and ticks are known to lead the flocks all over the world. Actually, in Europe as well as in Southeast Asia, ticks are very important to all the mixed species flocks. And here in particular Western Himalaya, the low elevation flocks are largely led by what I think is grey-headed canary flycatcher, because they're always active, calling their loud call. You can hear them from here actually in winter. They will be always calling in the salt forest. So I think they're important.
Yeah, and other important thing is with respect to resources, like when you compare resources with respect to elevation, so they are almost same, but the flocks change.
Right.
So do you think other than resources, the forest configuration and forest complexity plays role in flocking? Because at lower elevation you will have a large number of trees. So are these flocks mid canopy, top canopy, or low flocks? So if they have that average height, maybe when you go up elevation to the Masuri, so that height range of those trees, the number of availability of those trees probably decrease, which results in more flocking intensity as compared to the lower elevation ranges. Probably that may be another factor to look at the forest configuration change, because if the arthropod availability remains the same, but the configuration of the forest changes, that may have an influence on number of flocks of flocking intensity. Probably if you have some that particular data set, and probably you will come to know. Otherwise, it was really interesting, and congratulations.
Thank you, sir.
Pranav,
Hello, sir.
it was a very good presentation, and I really enjoyed listening to your talk. I have a couple of questions.
Okay.
Towards the end, you had said that there's not any difference in terms of resource availability,
Yeah.
right? But what about the disproportionate sort of effect of use on that resource? For example, what about the densities of birds or flocks as you go from lower elevation towards higher elevation? Because the effect of availability, okay, or use is very relative, as you go, and your study itself says that there is a difference in the number of flocks, right? Did you mention that? Yeah. So definitely, if the availability is same, but there is a difference in the number of flocks, then that effect has to be sort of accounted for.
The differences in the number of flocks is largely due to the flocking period. So when on the onset of winter, when birds move down to low elevations, they stay in the low elevation just for the few months, let's say mid-March, and they start moving up slowly on the onset of summer. So the sampling period itself was very low for the low elevation. So even though I sampled further in the April, I saw the resident birds, but they did not participate in the flocks. They already had started breeding, and they were moving on their own. And in terms of availability of or the densities of birds, the densities were definitely high in the low elevations. But since I use null models, I control for their availability. I control for their relative abundances. And the interpretations were made by taking considerations of their availability and comparing them with the observed values. So even though the densities were different across the elevations, it does not have any effect on the interactions per se, because they were compared in response to, in the light of their availability.
Okay, yeah, to some extent I agree with you that the null model simulations will probably help you in sort of teasing that apart, but I would like to, sort of you to look into that aspect of, the variation in the densities of birds, and also there will be definitely variation in the detections of birds, and whether that has any effect. And if it does not, then it's really good to, at least confirm that with that fact that it actually does not. So null model simulations are good. They help you and understand things much better, but definitely, they also have certain biases in that. The next question is, you know, so the question is to flock or not to flock,
Yes.
right? So in terms of flocking, is there is there a lead species or a group of lead species that you found that were really the first ones to sort of initiate the flocking in these two sites? Were they different in these two particular sites at lower elevation as well as high elevation?
Yes. So I'll speak more about low elevation plots where there is species called as cinereostid, which is common here also in the campus. And it was always in the plots in winter. But the day I stopped seeing cinereostid around, the flocking had already disintegrated. So I think cinereostid is a very important species for the low elevation plots, along with other species like canary flycatcher, white-throated fantail. They always call, they are always active than other birds, they are always ahead of other birds. Whenever they cross from one side of the road to other side of the road or one tree to another tree, these species are always ahead. But since I do not have data on who leads the flocks every time, I